Okay, so just to kind of go over a few genetic disorders, just a few. I mean, there's a lot of them. So I just kind of cherry picked a few to kind of go over. So we know how mutations work. We discussed mutations, point mutations and frame shift mutations. When you just change one nitrogen base, maybe you change a T to an A or a C to a G or something like that, you end up changing the mRNA. Nitrogen base, which can then change what amino acid is coded for, and that can change the entire protein. And so we know that in order for proteins to work efficiently and correctly, then the genes that, or the DNA that code for them have to be correct. And when they're not correct, we end up with a protein that does not work correctly or malfunctions or just doesn't exist. So let's look at situations where you have a mutation in the DNA, which translates to a mutation in the mRNA, which then translates to a misfunctioning protein. The first one, and we've discussed this one before, but sickle cell anemia. Sickle cell anemia is a situation where you end up with a point mutation in the DNA that codes for the hemoglobin protein. The hemoglobin protein is a protein that is on the red blood cell. It is actually kind of inside of the cell membrane, and its whole purpose really is to hold on to oxygen and to carry that oxygen throughout your body, because you know your red blood cells travel through your circulatory system from your feet to your head and back. Now they deliver oxygen and glucose and all sorts of nutrients that you need in order to survive. That is what they do. They're the delivery system. Oxygen being obviously the most important. So hemoglobin holds on to that oxygen. In order to do so, hemoglobin has to also hold on to iron. As it holds on to iron, that is what gives your red blood cells that nice red color, because oxygen plus iron makes red. That's just how it works. Now, you'll learn that in chemistry. So whenever iron is oxidized, it does turn red. When you have that situation, hemoglobin is so well embedded in that cell membrane that it actually determines the shape of the red blood cell. And red blood cells actually kind of look what we call discoid. When I say discoid, they kind of look like donuts, but not full with a full hole in the middle. They just look like donuts with kind of an impression in the middle. That's how they normally look, and that's because of how the hemoglobin is embedded in the plasma membrane. That's what gives it its shape. So hemoglobin is very, very important. Again, it's a protein embedded or inserted into the cell membrane of red blood cells, and it carries oxygen and iron, and it determines the shape of the red blood cell. That having been said, individuals with sickle cell anemia have a point mutation in their DNA that then codes for a misfunctioning hemoglobin protein. That misfunctioning hemoglobin protein, number one, does not carry oxygen the way that it should. And as a result, it also doesn't carry iron in the way that it should. So you can see this particular cell is not even as red as the other cells. It's actually a little bit more orange. So it doesn't carry as much iron and doesn't carry as much oxygen. And since it is malshaped, it's going to misshapen the cell itself because it's malfunctioning. And so, like I said, hemoglobin carries oxygen, carries iron, and it determines the shape of the cell. And so therefore, this cell, A, doesn't carry oxygen very well, B, doesn't carry iron very well, and C will be misshapen because of its malfunctioning hemoglobin proteins. And so that's where you get that sickle shape, which comes from a type of farm equipment called a sickle. You can Google that. But that's the reason why it's called that, is because that's what it looks like, a sickle. All right, so now, how does this happen? It's actually a form of evolution, which sounds really strange, I know. But in places like in tropical parts of Africa and in parts of the Caribbean, as well as tropical parts of India, you have a particular disease called malaria, and that malaria is caused by a nasty little parasite. That parasite is called Plasmodium, The plasmodium lives inside of mosquitoes. And so when the mosquito attaches to someone and starts sucking out their blood, then it sometimes delivers that plasmodium into that person's bloodstream, and that plasmodium then gives that person malaria. Now, malaria really does attack the liver and eventually kills people. But what we've noticed is that the individuals who are heterozygous for sickle cell anemia, in other words, big A, little a, they somehow have some sort of protection against malaria, which is very bizarre. Now, bear in mind, sickle cell anemia is homozygous recessive, so it's little a. Like, it is not at all it's not dominant, it's recessive. And so these individuals do not have sickle cell. Okay, they carry it. They're big A, little a, so they carry it, but they don't have it. They don't have the disease. So not only do they not have sickle cell, but they also have a protection from malaria. So you end up with populations of individuals that are selected for this heterozygous condition, because the individuals that are homozygous dominant, big A, don't survive malaria. They don't live and they don't survive malaria because they don't have the heterozygous condition. And the individuals who are homozygous recessive, little a, they have sickle cell, and so they don't survive. So you end up with large populations of individuals who are heterozygous, big A, little a. And so those individuals are typically going to be people from tropical places, and that's why you see a lot of Black people with sickle cell. So a lot of Black people are going to have sickle cell because our ancestors are from those areas, from the parts of Africa that are tropical and subtropical. But you also see a lot of sickle cell in like Indian people from India, because a lot of those places have malaria, and as well as in the Caribbean. So darker colored people, darker melanated people tend to be the ones that have sickle cell anemia. That's kind of how that works. But that is a genetic disorder, and it's a serious genetic disorder. People often do die of this. We have a lot of treatments now for sickle cell anemia, so they do live longer. I mean, you could live to be, 50, 60, 70, 80 or whatever, but you're not going to live the same kind of life that everybody else does. Like, you're not going to go out in the sun and hang out because you don't have as much oxygen as everyone else. You're going to get more tired and dizzy faster than everybody else. So this does change your lifestyle quite a bit. Cystic fibrosis, this one's really fatal. This one right here, honestly, you can't survive this past 30. This is going to be common in Caucasians. And so what happens with cystic fibrosis, is that you end up with an individual with this recessive disorder. It is a mutation. It's a frame shift mutation in the DNA. And what happens is that it codes for a sodium potassium pump that doesn't work. And we talked about sodium potassium pumps. We said that was an active form of transport, cellular transport. And that sodium potassium pump that's malfunctioning is actually in the cell membrane of lung cells. And since it does not pump out enough sodium, what ends up happening is you end up with a nasty little mucus membrane around those cells in the extracellular fluid outside of the cell. And that mucus membrane, that mucus builds up and builds up and builds up. And remember, this is in the lungs. And so these individuals have trouble with breathing because they have mucus all around their lung cells. And so over time, that starts to clog up the airways, and it starts to block up ducts, and even in the liver. And eventually that person does kind of suffocate to death. The only treatment for this disease is a double lung transplant. That is the only treatment, that's the only cure for this disease. Okay, so pedigrees. A pedigree is a family history that shows a trait and how it's inherited throughout the generations. And so let's say, for instance, that in your family, you've got someone who's a dwarf, maybe your uncle or your cousin or what have you, and you say to yourself, boy, I tell you, I don't really want my child to have to live that life. And you're ready to have children, but you're not sure if you carry that dwarfism gene. Remember, these disorders are recessive, and so they hide in families. All of them hide, and so you may be completely normal, but you may have a child that has dwarfism or sickle cell anemia or cystic fibrosis or Tay-Sachs disease or any of these other very, traumatic disorders. And so you may want to consult what we call a genetic counselor that will create a pedigree for you so they can calculate and determine your chances of passing on those traits. So here are the rules for a pedigree. You've got a shaded box, and that's going to be a male that has the disease, and a shaded circle is a female that has the disease. Then you get to the clear. Clear box is normal, does not have the disease. Normal male, and normal female is a clear circle. A carrier is half shaded. Now, you're not always going to get a pedigree that has the carriers half shaded, because that might be part of the question. They might be asking you, hey, what is the genotype of this person? And they're not going to give you the half shade because you can figure that out because that's a heterozygote. So sometimes you get the half shades and sometimes you don't. But these guys are carriers or heterozygotes. Okay, so these guys are affected. These guys are unaffected. So boy affected, girl affected, boy unaffected, girl unaffected. So completely normal. And then these two are going to be heterozygotes, which is going to end up translating to being normal, but they can still carry it and pass it on to their children. So first thing you want to do when you see a pedigree, first and foremost, recognize that over here you have 1, 2, and 3. These are the generation numbers. So this is grandma and grandpa, this is mom and dad, and this is you, your cousins and your sisters, right? So these are your generations right there. This, when I have a box and a line and a circle, the line shows a mating. So these two individuals mated, okay? And then I have a line that goes down and a line that goes across. These are their children right here. This person is not their child, and this person is not their child. This person is their child's mate or husband right here. And so is this individual, okay? Because this starts a whole new pedigree right here. So one of the first things you want to look at to determine what type of trait you're looking at for a pedigree is whether it's autosomal or sex-linked. Now, we discussed autosomes and sex chromosomes earlier in the previous video when we did sex-linked. Autosomes are going to be those chromosomes that code for regular body stuff. So if we're talking about your somatic cells, like the cheek cells, like your cheek cells, skin cells, nerve cells, blah, blah, your regular body cells, they have 46 chromosomes. 44 of those chromosomes are autosomes. They code for regular body stuff. Can you roll your tongue? Do you have hitchhiker's thumb? Do you have a widow's peak? Blah, blah. The other two are your sex chromosomes. They determine your gender. So you're either XY and you're a boy, or XX and you're a girl. And so when we're looking at these pedigrees, we're determining whether or not this is a trait that is on the autosomes, or is this a trait that is sex-linked on the X chromosome? Easiest way to determine that is to look at all the affected individuals and determine what their sex are. So these two individuals are male, and then I've got 1, 2, 3, 4 that are female. Now, if you remember from our sex-linked video, the majority of individuals that have X-linked diseases are male. So if it is a sex-linked trait, the majority of shaded shapes are going to be boxes. They're all going to be boys. But in this one, that's not the case. I've got 4 girls and 2 boys. Okay, so I know that this is autosomal because there's more girls than there are boys. Because in sex-linked, there'd be more boys than there are girls. Okay, so that's one way to determine whether or not you're looking at a sex-linked or an autosomal trait. So we've determined that this is autosomal. This is not sex-linked, okay, because there's only 2 boys, but there are 4 girls. Now, dominant or recessive. Recessives hide. I just said that, recessives hide. So you can have 2 individuals that are completely normal. You see these 2 individuals that are normal, boom, boom, normal, but they can have an affected child. When you see that, when the two individuals, the two parents, mom and dad, are normal, but they have a child that's shaded and is affected, then that is recessive. That is a recessive trait. That's the only way that can happen. There's no way that these two individuals could be dominant and I'm sorry, there's no way this person could be dominant and neither one of the parents have a dominant. That doesn't work. That doesn't work. If you put it like if I have big B, and big B, little b, these guys would be shaded because they both have the trait. So this individual right here, if this person was big A, they would have to have gotten that big A from either dad or mom. Or big A, little a, again, would have to have gotten one big A from mom or dad, but neither one of these have it. So therefore, this must have been recessive. Both parents are going to be heterozygous. Okay, so both parents are heterozygous, big A, and big A, little a. And that 1/4 little a is right here. Same thing here. Okay, same thing here. These two are normal, but yet they have two children that are affected. So that is definitely recessive. So we've decided that this is autosomal and it is recessive. All right, heterozygous or homozygous. Well, this gets a little confusing. This individual, in order to be little a, so each one of these individuals has to be little a. So that means that mom and dad here have to be big A, little a, and big A, little a. That's the only way that this child could have gotten a little a, because this parent had to have one little a and this parent had to have one little a. Same thing here. This parent had to have one little a, this parent had to have one little a to give two. So this father gave a little a to the son and this mother gave a little a to the son. So this son is little a. Okay. Easier here. So this mom had to be big A, little a to cross with a little a to make a son that's little a. Couldn't be big A because a big A crossed with a little a would make all heterozygotes and that would be normal and he would not be shaded. So she has to be a heterozygote. So that's how you have to look at this. So this is actually a pedigree for an autosomal recessive disease called albinism. That is characterized by a person's inability to produce an enzyme that's necessary to make melanin, whether that's eumelanin or phagomelanin. Remember, eumelanin is going to make that dark colored skin like Black people have or Indian people have, people who are who have ancestors that come from places that are closer to the equator. And then, of course, phagomelanin is going to be a lighter colored melanin, but it's necessary for the production of vitamin D. That having been said, you need a particular enzyme to produce either one of those types of melanin. And so those individuals don't have that enzyme because they have a mutation in the DNA that codes for that enzyme and causes that enzyme not to be produced. Remember, enzymes are proteins. So this protein here does not get produced because of that mutation. Now that makes them very light. They're albinos, and so obviously there are a lot of things that go with that as far as how they have to live. But anyways, remember, I've got 1, 2, 3 generations. So grandma and grandpa are normal, but they're heterozygotes. They're carriers. They have to be in order to have a child here, right here, who's little a, and a child here who's little a. Like they have to be normal. They have to be heterozygotes. And then this daughter here has to be a heterozygote as well as this man here. These two individuals have to be heterozygotes in order to make a baby here that's little a. Both of these individuals have to be heterozygote, big A, little a, in order to make two children that are little a. Okay. Now this one's a little bit different. This child is little a, and the woman can't be big A, because if she were big A, she's homozygous dominant. A big A crossed with a little a makes all big A, little a's, but that's not what happens here. You end up with a little a. So this individual has to be big A, little a. She has to be a heterozygote. So she donated one little a, and he donated one little a, and so therefore you have a son that has little a, little a. These are homozygous recessive, autosomal homozygous recessive traits. So let's practice. We're going to do an X-linked. Remember that if it's an X-linked, you're going to see more shaded boxes than circles, way more. In fact, you may see all shaded boxes because that's just not, having a shaded circle is rare. A man and a woman marry. So here's my man right here. Here's my woman. Now, I didn't half shade her, but she's a heterozygote. Okay, so you can half shade her if you want. They have 5 children. They have 2 girls and 3 boys. Okay. She passes a gene, so she is a heterozygote. So she's X big H, X little h. So she passes the gene to 2 of her boys. So this child is X little h, Y, and this child is X little h, Y. And 2 of her boys who died in childhood. Hemophilia is a nasty little disease, and so yeah, it's entirely possible to die from it. So now you end up with 1 boy and 2 girls. One of her daughters is a carrier, and so when she gets married, she ends up with a child with hemophilia, which makes sense. The only way a boy is going to get hemophilia or have hemophilia is if his mom is a carrier. It doesn't matter what his dad is, because his dad gives him a Y chromosome. The mom is the one that gives him the X. And so the mom here donated the X little h to this child, so he's X little HY, and so he's a hemophiliac. Over here you have a non-carrier daughter, and one of her sons, so she's X big H, X little h, and one of her sons, he's X big H, Y, marries a woman who's a carrier. And it asks you have twin daughters, what % chance that each daughter will also be a carrier? Okay, so she's a carrier, she's X big H, X little h, but he's X big H, Y. So we're wondering, what are the chances of the children, the girls, the twins, being carriers? All right, so let's put some of these genotypes on there, because I'm saying them and you're like, what? All right, so grandma here was a carrier, X big H, X little h. Remember, she's XX, she's a girl. And then here's dad, X big H, Y. All right, now mom is normal, dad is normal. She's a carrier. Really, she should be half shaded, but okay. She's a carrier. She donates her X little h to this son and to this son, all right? So you end up, and she donates an X little h to this daughter, but dad comes in clutch with that X big H. So this daughter's normal, but she's a carrier. These two, because mom's little h there, they're hemophiliacs. They end up dying. This little boy gets lucky and he gets the big H from his mom. And this little girl gets super lucky. She gets the big H from mom and the big H from dad. So she got super lucky. This daughter, the carrier daughter, marries a normal man. And this daughter right here, the non-carrier, totally normal, big H marries totally normal man. This daughter here, who's a carrier, ends up passing the little h to her son. So he ends up being hemophiliac because that little h is all he has on his X. He only has one X. And so dad gives him the Y. This son's totally normal because he ends up getting mom's X big H. This daughter is very, very lucky because she gets a big H from mom and a big H from dad. So she ends up getting kind of lucky in that respect. This child right here, this individual right here, is an individual that married one of their children, and she just so happened to be a carrier right here. So she just so happened to be a carrier. And then this individual, this man, is only possibility he could have is X big HY, because that's all his parents have, are X big H's. They're all normal. Again, they're all normal. That's the only thing they could have. again, the parents are normal. So all of these children right here are going to be big H's all the way across because parents are normal. But this kid right here ends up marrying a woman who's a carrier. And so the question becomes, what are the genotypes of their twins? What could they possibly be? So what is the chance that their twins, these two girls, are going to end up being carriers? Because they could end up being X big H, X big H, or they can end up being X big H, X little H. So what are the chances of that? So we put that in a Punnett square. Here's mom, she's a carrier, she's got that little H, and here's dad, he can only have the big H. So out of the girls, because they're only girls, we're not even going to look at the boys, they don't have any boys. So out of the girls, they have a 50-50 chance of being carriers. Okay, so going back and putting those genotypes back. So there's only a 50-50 chance, there's only a 50-50 chance because one girl, or one chance is X big H, and the other one is X big H, X little H. 1 out of 2 is 50%. So the answer to that question is 50%. Okay, so really, these take practice. You have to look at them for long periods of time. So you need to practice, 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 practice.